All right. So today is the last step in cell communication, and that last step is response. So we talked about the different types of receptors: G protein coupled receptors, receptor tyrosine kinases, and ion gated channels. Then we talked about transduction yesterday, with signaling transduction pathways. And then today we are finishing it up with response. On Monday, we're going to talk a little bit about how apoptosis, the process of apoptosis or programmed cell death, depends on these signaling pathways. And then, check. We'll do like a little activity to kind of loop this whole process together. Hopefully, those will both be done on Monday. And then we move into cell cycle mitosis stuff, which will take us through next week. It's going to be a little bit content heavy, but I'll try to put stuff in there to kind of break it up. Just kind of get to the halfway point by the halfway point. But today's lecture is relatively short compared to the other ones, so I'll probably have some time just to vibe at the end of class today. Before I get started, anybody have any questions, comments, concerns, fun anecdotes? All right. So today is cell communication day 4. We are all about response. So, like I said, we talked about how receptors receive signals. We talked about how those receptors function to trigger the next step in the process, and that next step being a signal transduction pathway of some sort, whether that be a relatively short phosphorylation cascade, a series of second messengers within that phosphorylation cascade that kind of triggers one thing to move, another thing to move, another thing to move, like a weird cellular Rube Goldberg machine. But the whole point of receiving a cell signal is to trigger a cell response. Now, most of the time, cell signaling leads to regulation of transcription or cytoplasmic activity. So, cell signaling leads to regulation of transcription or cytoplasmic activity. Or cytoplasmic activity. Many signaling pathways, their entire goal is to regulate protein synthesis. Many signaling pathways ultimately lead to the regulation of protein synthesis by turning genes on or off. I need to fix my handwriting. By turning genes on or off. And I put quotations around on and off because it's not always an all or nothing situation. In biology, rarely ever is something black or white. So odds are, if we are turning things on or off, We are just dramatically increasing or dramatically decreasing the rate at which something happens. So odds are a gene is being transcribed, but just not at the pace a cell needs. So the cell will receive a signal to kind of ramp up that transcription factor, not necessarily just like flip a switch, turn it on or off. But sometimes they regulate protein activity. So when they're not turning on or off transcriptions, signaling pathways can also regulate protein activity or regulate protein activity. This is like opening or closing ion channels. Or increasing or decreasing metabolism. Metabolic enzymes, all of the enzymes that we talked about with photosynthesis and cell respiration, anabolism, catabolism, can all be affected by signals received via these signal transduction pathways. So, let's see. How do I want to bridge this gap? A cell response, most of the the entire point of cell signaling is to regulate what the cell does. But in order to regulate what the cell does, we have to regulate how the signals are received. So the essentially the sorry, my brain's not back yet. The overall theme of today is regulation of response. That's the whole goal of cell signaling. So, like I said, it's more complex than just turning things on and off. It's not like these signals just go around flipping switches everywhere. There is a lot of complicated aspects. We're going to talk about 4 aspects of regulation. Now, this isn't all of what goes into regulation, but it covers pretty much all of our bases. And that is what we're going to talk about today. What in the world? Okay. Number 1 is amplification. A big component of cell signaling is being able to amplify that signal in order to ensure the entire cell responds to it. The second is specificity. When hormones are released into your bloodstream, those can act as signaling molecules, right? So if it's just released into your bloodstream, why is it that certain cells respond to it and other cells don't? That kind of stuff. Number 3 is the efficiency of response. How can we be sure that we are not wasting signaling molecules or wasting the proteins involved in the signaling transduction pathway? Cells are like little machines, and while they get the job done, they aren't necessarily always the most efficient systems. And finally, termination of the signal. Once something gets turned on, how do we turn it back off? If we've got signaling molecules loose in the body, how do we tell the cell that, okay, you don't need to be having a certain response anymore? So that's the goal of the day. We're going to just work through them one by one. Nothing too crazy. I think I've got one little picture for you, but overall, nothing too bad. So, starting with amplification. It's really big. Starting with amplification. So, the signaling pathways that we talked about yesterday, those phosphorylation cascades, are what amplifies a response. How we get one signaling molecule reaching the outside of the cell to lead to a series or a cell-wide change. How can one molecule affect the entire cell? And that has to do with our signaling cascades. Elaborate signaling cascades amplify a cell's response. This is like a little dot. If you remember our signaling cascade from yesterday, we had a relay molecule that turned on protein kinase 1, which will then activate protein kinase 2. And some of them have 3, 4, 5, or 6 proteins in this step. So, at each one of those steps, the number of activated products is greater than the preceding steps. So it's essentially like a pyramid scheme, right? But going down instead of up. So at each step, the number of activated products is greater than the preceding step. So if an active protein kinase 1 if an active protein kinase 1 activates 2 protein kinase 2s, and each of these protein kinase 2s activates 2 protein kinase 3s, and so on and so forth, we can get a pretty big cell response triggered by just one signaling molecule. And in that way, it can amplify a cell, so we don't need a million signaling molecules in order to turn on what we need to turn on for a cellular response. So that's signal amplification. Any questions? The next is specificity of cell signaling. Specificity of cell signaling. And with this comes the coordination of the response. So, This step specifically boils down to the fact that different cells have different collections of proteins. Different cells have different collections of proteins. Now, so far, what we've learned is that the response depends on the receptors, which is still kind of true. Whether or not a cell can respond to a signal does depend on whether or not they have the equipment to detect the signal in the first place. If a cell doesn't have a receptor to respond to a certain signaling molecule, it won't have a cellular response in response to its presence. But another way that cells can increase how specific a response is by the difference in the proteins that respond to the receptors. So we can have two cells with the same receptor protein, but differences in the proteins in the signaling pathway mean we get different responses. So, let's see. What example can I give here? Caffeine, right? So, I drank this giant thing of tea this morning, almost all of it. The way my heart cells react to that caffeine is going to be different than the way my brain cells react to that caffeine, right? So, they might have the same receptor, because why waste the equipment to respond to a molecule in our DNA, right? We have a limited amount of genes. If we have the genes for a receptor that accepts caffeine molecules, why have two different ones for two different types of cells? So, heart cells, brain cells, both have receptors to react to caffeine. But our brain cells respond in a more electrified potential kind of thing, and our heart cells respond with like a pace increase, right? Different responses, different types of cells, same signaling molecule. So, yes, the response depends on the receptors, but we can also have the same receptor protein, but differences in the proteins in the cascade will give us different responses. Your liver cells react differently to alcohol than your brain cells do. Same signaling molecule, different response. So that's another way our cell pathways can get specific based on different proteins. Another way is branching and interactions between pathways. So if sometimes one pathway will happen, but if a different receptor receives a different signal, it can trigger a response that stops this one in its tracks. or this one can have like a safety backup. So if pathway A happens and it's triggered by a signaling molecule, and pathway B happens and it's triggered by a signaling molecule, but let's say and both of these need to work together to get a job done, right? But if pathway B doesn't have its signaling molecule, now we don't get our cellular response because we don't have this half. But we could have a branch go from this pathway to this pathway and be like a safety backup trigger, just in case the second signaling molecule didn't happen, but we still need our cellular response. So branching and interactions between pathways are important to regulation and coordination of a cellular response. Branching and interactions between pathways are important to the regulation and coordination of a response. So, responses could be regulated by amplification. Responses can be regulated through specificity, and they can also be regulated by efficiency. So, number 3 is signaling efficiency. So, how does a given protein kinase find its substrate? Oh my goodness, I hate when I do that. So we got a signaling molecule. It hits a receptor. The receptor activates protein kinase 1. How on earth does protein kinase 1 find protein kinase 2? We only have a given concentration of them. It's not like they've got like GPS locators, and the cell to a signaling molecule is actually pretty big. So, how does one protein kinase find its substrate? In many cases, say many because, it's not all of them. In many cases, the efficiency of a pathway, so how quickly a pathway can get done so we're not wasting signals, is increased by the presence of scaffolding proteins. So these scaffolding proteins help kind of hold things in place and connect everything to where they need to go. So in many cases, the efficiency of a signaling transduction pathway efficiency of signal transduction is increased by the presence of scaffolding proteins. Scientists were measuring a cellular response in a certain cell or outside of a certain cell. They got the enzymes they needed to artificially create a pathway. They measured the rate of efficiency, and then they added more scaffolding proteins to the mixture. And the signal got transduced a lot quicker, or they inhibited the synthesis of the scaffolding protein. They took the gene for the scaffolding protein, yanked it out of there. Cell can't make it. Signal transduction decreased in efficiency by a ton. So, well, what are these scaffolding proteins? Scaffolding proteins are large relay proteins to which several other relay proteins are simultaneously attached. So, these are large relay proteins to which several other relay proteins are attached. So, this is the thing we're going to draw out. We've got, just like before, our cell membrane. We got, backwards. We got the outside of the cell. We got the inside of the cell. We've got our receptor. When that receptor binds to a signaling molecule, it activates. That we know. But instead of just one relay protein attaching to this, we can have something called a scaffolding protein. So, draw it like this. This is our scaffolding protein. This scaffolding protein can have many different signaling molecules attached to it. So, when the scaffolding protein attaches to the receptor, it can relay the message threefold. Now, these can all three be the same, so we're going to just kick off three of the same pathway. But more often than not, we've got like two of the same and one completely different, or three completely different types of relay molecules on that scaffolding protein. So one signal can kick off multiple responses. Yes. Yeah. We're just going to call them relay molecules. They can all trigger the same response, they can all trigger different responses. But it is more efficient for one receptor, one signaling molecule, to send off 3 responses than have 3 receptors send off 1. Let's see. Let me get this back to what I need it to be. So, that's a scaffolding protein. They can behave in a couple different ways. Some scaffolds are permanently, some scaffolds permanently hold networks together. A little messy. A big example of this is in the synapses of our brains, right? We want to make sure that signals get from the axons of one nerve cell to the dendrites of the other, right? We want to make sure that our neurotransmitters get from point A to point B as quickly and as efficiently as possible. So in our synapses, we have these scaffolding proteins that help connect the two ends of our, or the ends of one neuron to the beginning of another neuron, so our neurotransmitters know where to go. So it's not just random diffusion, we have a path to follow. And in other cases, our scaffolding proteins directly activate these relay proteins. So instead of just attaching, binding, and releasing the relay molecules, they can actually activate the relay molecules. Ignore that period. Some directly activate relay molecules or proteins. So that does get right down. All right, and finally, this is taking longer than I thought. We have termination of the signal. Just like we got to start a signal, we have to be able to stop it. If caffeine triggered a response in one cell, we want to be able to stop that response once the caffeine's gone. Yes? So, inactivation mechanisms are essential for any signaling pathways. Inactivation mechanisms are essential. Inactivation mechanisms are essential. So, in order to remain capable of responding to signals, each change in a molecule must only last for a short time, right? So if a signaling molecule changes the shape in the receptor has to very quickly be able to reverse that change. In order to remain capable of responding to signals, each change in a pathway must only last for a short time. that signaling molecule that initially triggered that receptor has to dissociate fast. So if there is another signaling molecule like right around near, it can bind and we can continue to propagate that signal. The ability to receive signals depends on reversibility. We'll start by that. So, a cell response only occurs when the concentration of receptors with signaling molecules, so the concentration of activated receptors, is what triggers the cell response. If not enough signaling molecules are attached to our receptors, the cell response doesn't happen. When that cell response doesn't happen, when those ligands dissociate, that is when the cell returns to inactivity. So one last little bit. Let's see. Cell responses. Ooh, that's hellacious. Okay. Only occur when the concentration of activated signaling molecules or activated receptors is above a certain threshold. If not, return to inactivity. Ready to receive. So, and I got a little messy at the end. If these changes aren't reversible, we can't propagate new signals. As you continue your education through cellular responses, you will learn more and more that everything depends on concentration gradients. So we can have a few activated signaling molecules, but if we don't have enough, nothing will happen. Same thing with how neurons work, same thing with how development works. Concentration gradients essentially run cell processes. So if we don't have enough signaling molecules, so if whatever signaling molecules our receptor tyrosine kinase releases, if there's not enough of them, even if there's some of them, we won't get our cellular response. Anybody have any questions? All right. There is an exit ticket on Canvas. You have like 5 minutes to fill it out. If you have any questions, please let me know.